Okay, so right now we will be discussing FACS. Okay, what is the full form of FACS? FACS stands for Fluorescence Assisted Cell Sorting. Okay, FACS stands for Fluorescence Assisted Cell Sorting. So what do you mean by Fluorescence Assisted Cell Sorting? This means that you will have to differentiate cells on the basis of fluorescence. That's it. You will have to differentiate cells on the basis of fluorescence. Okay, so we have already discussed what is flow cytometry. We have already known what is flow cytometry, but today we will be discussing what is FACS. Now listen, there is a big difference between flow cytometer and a FACS machine. Why? A cell sorter is not equal to flow cytometer or flow cytometers. A FACS machine or a cell sorter is not equal to flow cytometers. Suppose you are given a machine and you are told that in this machine you can do FACS as well as you can do flow cytometry. That means this is actually not a single machine. It has got two functions. That means it has got two different systems for flow cytometers and FACS. These are actually two different machines which is jumbled into one particular jacket. That's it. Whenever you are told, in most of the regular practices, it is already said in lab that whenever we are actually supplying or we are actually taking any flow cytometers, we are already taking into account that it also contains FACS facility. But it does not necessarily mean that all flow cytometers will be fluorescence assisted cell sorters. Okay. Now, why we put FACS machine or flow cytometer machines in FACS machine? That means why these two machines are put together. Have you seen any radio with a television type thing? No, right? Have you seen any computer with a typewriter type thing? No. Have you seen any fan with AC type regulation? No. Then why these two things are put together? There is single reason for it, because the starting point of flow cytometer and FACS are same. That is, the fluidic system remains the same. The starting point of FACS and flow cytometer is a fluidic system. This is the only similarity between the FACS and flow cytometry machine or flow cytometer. All the other compartments or components are different. Okay. Now today we will be discussing FACS in a very lucid manner. Okay. Listen, when we are talking about FACS, you should understand that it is actually a continuation of the previous lectures. So you should understand that whenever we are talking about FACS, that means we will have to talk about hydrodynamic focusing. We will have to talk about hydrodynamic focusing, which we have already discussed. That means here there is a fluidic system, right? This is the fluidic system. Okay, and this is your chamber where you will have to inject the specimen. This is the tank or the beaker where you will have to put your specimen. Okay, now listen with all your attention. We have already discussed that with the help of hydrodynamic focusing, we have already created a streamline flow of any fluid, right? We have already created a streamline flow. But when you are trying to know FACS or fluorescence assisted cell sorting, we will have to add many additional compartments here. What are those additional compartments? Listen. You should know one thing, that whenever you are talking about FACS, this particular compartment actually plays the major role. Fluidic system actually plays the major role. But now what I have told is, when we are using flow cytometer or when we are using FACS, then actually we are using the same fluidic system. That means suppose when we are using flow cytometer property of this particular machine, then the additional property of the FACS machine is turned off. What is that additional property? Now listen. With the help of hydrodynamic focusing, we already know that a streamline flow of your injected cell suspension is created, right? With the help of hydrodynamic focusing. I am not going into details because we have already discussed it. But there is an important change in this. What is that? That suppose these are your fluid which you have injected. Okay. Now, two components I will add here. Two components. One is a charged tube. Another is a simple vibrator. That's it. That means I will be using a charged tube here, okay, a charged tube here, a charged tube, and another I will add is, another thing what I will add here is a vibrator, okay, a vibrator. Now, just because of the addition of these two things, the entire thing has changed. Just because of the addition of these two things, the entire thing has changed. Now listen with all your attention. Suppose, now we'll talk about how the, changed fluidic system of a flow cytometer acts when it is converted into FACS machine. This is a changed fluidic system. Now listen. You know that suppose this is a tube. Suppose this is a tube. Any fluid coming out of the tube have a tendency to become turbulent at this point, right? Right? Wrong. Listen. At this point, they will be streamlined. No doubt about this. But there arises a problem when this thing will happen. Suppose I draw the similar tube here. I draw a similar tube here and I add a nozzle to it. The addition of a nozzle creates an additional problem. What is that? That means when a fluid is, flowing from this side to this side, then the fluid layer which is coming here, which is actually following this path, is not actually, obstructed by any layer, but the fluid layer which is actually following this path has been obstructed by this thing. That means this fluid layer will now reflect its path. That means it will now interact with this path and there will be turbulence. And that's why due to this turbulence, the fluid which is coming out will have a tendency to form droplet. And probably you are now hearing this thing, right? Probably you are now actually listening and you are actually now learning this thing, but this thing was actually, this whole phenomenon was actually discovered by Rayleigh, by Lord Rayleigh. Okay, who said this thing, that whenever a nozzle is introduced into a streamline flow, then the particular fluid which was undergoing previously streamline flow will have a tendency to break into droplets. Clear? Okay. And the funny part, Rayleigh started this whole thing which you are actually now reading in 1800. He actually discovered this in 1800. Okay. Now, okay, up to this you have understood, right? So now listen with all your attention. What is our motto in this fluidic system? Suppose this is a flow cytometer fluidic system. Now we want to convert a flow cytometer fluidic system into a FACS fluidic system, or Fluorescence Assisted Cell Sorting fluidic system. What problem we will have to face, Listen. First, understand the objective of FACS. Listen. in a FACS fluidic system, we will make droplets. We will make droplet of this liquid which is flowing from here. Our main objective is to make a droplet, and in each droplet there will be a particular type of cell, and in each droplet there will be a cell. That means we will have to first form the droplets, and simultaneously we will, in each droplet we will have to insert one cell. This is our objective. That means we will have to break streamline flowing fluid into droplets, and in each droplet we will have to insert one cell. That's it. And each droplet should be charged, either negative or positive, or it may remain uncharged. That's it. Okay, this is our motto, or this is our objective. Now we will learn step by step how cells are inserted. We'll learn later, but at first let us discuss how these droplets are formed out of streamline pathway, right? Suppose we are following the basic fluidic system components, that is flow cytometer fluidic system. What will happen? Cells will come one by one, right? But when you understood the flow cytometer fluidic system, you should have known that when cells were coming, they were coming in a streamline flow, single, and each and every cells were aligned in a single column, right? But there was no droplet formation. Wait, I'm rubbing this thing. Listen. When you understood the flow cytometry fluidic system, you understood that when you studied the flow cytometry, you understood that flow cytometer fluidic system actually creates a streamline flow where one cells were coming at a regular interval, right? But here what we will do, we will actually tear these things and make a droplet out of them. That's the very basic difference. We will make a droplet. We don't want a streamline continuous flow. Okay, we don't want a streamline continuous flow. We want a droplet. How to do it? Now, before we question how to do it, we should understand why it is not possible in a normal fluidic system, right? Because in a normal fluidic system there is no nozzle. If you introduced a nozzle, then the flow cytometry machine then and there will be converted into FACS. And in a lab, you need both flow cytometers as well as FACS. That means you will have to invest 10 to 20 lakhs for flow cytometry, 10 to 20 lakhs for FACS. That means up to 40 lakhs you will have to invest for a single experiment. You are genius and your lab will be shut down within 3 months. That means if you are ordering a FACS or flow cytometer machine, that machine should contain a specific segment where it can be used as a flow cytometer as well as a FACS. How to do it? Listen, it's a very important part to understand. Listen, if we want to convert this streamline flow into droplets, we will have to agitate this nozzle. That's it. We will have to vibrate this nozzle at a very high frequency. That's it. We will have to agitate this nozzle so that fluid which is coming, listen, it's a very important part to understand. Suppose this is your fluidic system. This is your suppose fluidic system, okay? And this is the nozzle. This is your suppose exiting nozzle, this part. Okay, this is your nozzle. Now listen, here is your sheath fluid compartment. You know what is sheath fluid, right? Now listen. Suppose you are vibrating at a high frequency this particular inner fluid compartment. What is happening? Suppose fluids are coming from here, right? Now if you vibrate this particular tube system this side, then what will happen? This particular thing moves. This particular thing moves here. The new position of the fluidic, the new position of the compartment is now here. That means what? That means this particular fluid, or this particular flow of fluid which was coming uniformly will now interact with this wall and they will become turbulent. Then if I in another moment I will shift it from here to here, then this fluid will become turbulent, and due to this turbulence a nozzle like thing will be formed, and it was same, here was turbulency and here also there are two turbulent streams, right? And due to this turbulency what will happen is the single flow, the main flow which was actually made up of cells, the streamline flow which is actually made up of cells will be broken down into simple droplets containing one cell. Why it is containing droplet of one cell? Because already the streamline flow has actually told us that in every, the streamline flow allow us only one cell at a time, right? In a flow cytometry we have already studied how a streamline will only contain a column of cells, one at a time will exit, right? That's it. Okay, that's it. Now listen, there are basic points you need to understand. High profile physics is applied here. High profile physics. Listen, high profile physics is applied in every part of the FACS system. What are those? Listen with all your attention possible. Listen. Suppose this contains sheath fluid, right? Sheath fluid is flowing from here, right? Sheath fluid is flowing from here, and here is your cell suspension, right? Now listen, what is the role of this vibrator? You have understood, to create droplets. But my question is, why this particular vibrator vibrates? The answer is due to an electromagnetic wave or an electromagnetic disturbances, which is called piezoelectric effect. This is called piezoelectric effect. Due to piezoelectric effect, this nozzle actually vibrates, and this piezoelectric effect is nothing but an electromagnetic wave. Okay, and every electromagnetic wave has their own lambda. That means lambda means what? Wavelength. Have their own wavelength, right? Now listen, why I am saying this, listen. Suppose this is your fluid, and suppose this is the vibrator. Suppose this is the vibrator which is actually vibrating due to what? Piezoelectric effect. Okay, piezoelectric effect. What is piezoelectric effect doing? It is creating an electromagnetic wave having wavelength lambda, suppose. Now listen, suppose this is your fluid which is on the verge of breaking into a droplet. Oh my god, it is not a droplet. So these are your droplets. I am not drawing the cells right now. Okay, now what is the funny part? Unlike this diagram, there is more funny part to it. Okay, that is, the funny part is the distance between the two droplets is lambda. That is the basic thing you need to understand now. The distance between the two droplets is always lambda, which is equivalent to the wavelength of the electromagnetic wave. Clear? Okay. Now, when was the vibrator activated? In this portion. The vibrator was activated in this portion. This is the portion where the vibrator was activated, right? The vibrator was actually doing zzz, okay, right? That means what? That means the vibrator is activated here, not that type of vibrator which you are thinking, but these are good vibrators actually. Now listen. So when the vibrator is activated, the vibrator suppose is activated here in this portion, and the droplet breaks off in this portion. This distance is called break off point. This distance is called break off point. This distance is called break off point. Okay, that is the distance where the vibrator was activated up to the distance which actually is the point during which a droplet is formed. This distance is called break off point. Clear? This distance is called break off point. Okay, so we were discussing this. Now listen. So now each drop will have a charge. Okay, now now you need to correct one thing. This is actually not a break off point. Okay, I'm sorry, this is called drop delay. This is called drop delay, and this point is called break off point. This point is called break off point. That is the starting of the formation of a drop. Okay, now the distance between the two drops we have already discussed. The distance between the two drops is actually lambda. What is lambda? The wavelength of what? The wavelength of the electromagnetic wave which is produced by piezoelectric effect. Right? Now, point you need to understand again here is this, that droplet formation has a single condition. A droplet formation has a single condition. What is that condition? The droplet formation condition is equal to lambda equals to pi d. What is d? D is actually the diameter of each drop. D is actually the diameter of each drop. So this is the condition of droplet formation. Okay. Now, there are many points you need to understand again, that what is the exact velocity of each drop? What is the exact velocity head of each drop? The exact velocity head of each drop is equals to frequency of this electromagnetic wave into lambda of this frequency, into lambda of this electromagnetic wave. The velocity of each droplet is equal to frequency into lambda. Now, point you need to understand here again is this, that now question arises that how a drop becomes charged. How a drop is actually charged. That is the main question here arises. Now listen, there is a very important discussion here, that this was your compartment where you will have to put your cell suspension. This is your sheath fluid suspension. This was your vibrator, which actually follows piezoelectric effect. Now listen with all your attention. Here you need to understand that this nozzle is actually connected to a wire. There is a main difference between FACS, sheath fluid container, and FACS sheath fluid container as well as flow cytometer container, sheath fluid container. That is, they are connected to opposite charges. And what is the potential difference of this particular thing? that is 100 to 200 volts. Okay, so these two wires are now connected to 100 to 200 volts. Now listen with all your attention. When the drops are coming out, now you should understand that the drops are actually made up of water and electrolytes. Why electrolyte? Because we are putting here cells. No, we are not only putting here cell, we are putting here cell plus suspension, that is cell suspension. What is cell suspension? Cell which is solvated in a buffer, and buffer contains ions. So this droplet contains ions, and we are actually charging those ions properly, and therefore this whole droplet becomes positively charged or negatively charged or something like that. Okay, clear? Now listen, first you need to understand one basic thing, that who takes the decision that this particular drop will be positively charged, this particular drop will be negatively charged, this particular drop again will be positively charged or uncharged, this particular drop will be positively charged. Suppose, who takes this decision? First question arises in your mind, who takes this decision? Now actually you have taken the decision, which cell will take which charge. Actually you have already taken the decision. Now how it is done, right? Now listen, to understand this basic thing you need to again understand few basic things. Now listen, actually what happens is, okay, now suppose you people are dealing with three types of cells. Suppose you people are dealing with three types of cells. First one is the yellow one, okay, yellow one. Second one is the red one, and third one is the blue one. Suppose you people are dealing with three cells. Okay, now the third one will be the violet one. Okay, third one will be the violet one. I need to use the violet because I will be drawing the fluid with blue. Now listen, suppose at first this cell is coming in a streamline fashion, and due to these vibrations or piezoelectric effect, a droplet is formed. Okay, and in this droplet the yellow colored cell is present. The yellow colored cell is present. Now listen with all your attention. You need to understand that how, suppose this yellow colored cell this droplet containing yellow colored cell will take positive charge. Who is telling that this drop containing the yellow colored cell will take the positive charge? You are actually telling. How you know? Listen, here is your optic system, side scattered channel, forward scattered channel, all these things. Now listen, suppose this is coming here. Okay, so this is the droplet, and inside the droplet is a yellow colored cell. Suppose is an yellow colored cell. Okay, and a laser is interacting with the cell. Okay, now listen, when the laser is interacting with the cell, we know that how, by the interaction of this cell and laser, computer knows that this is the yellow colored cell, right? Computer knows, computer is now very much aware that this is a yellow colored cell. Okay, now listen, it's very important from your point of view to understand that how this cell becomes charged. Okay, so now we were discussing that, so suppose we are taking into account that this particular drop containing an yellow cell, okay, containing a yellow cell will have positive charge. Now who is telling this particular system that the cell containing the bubble or the droplet containing the yellow cell will have a positive charge? Who is saying that? We are actually instructing computer. How it is doing? Now listen, we are not going into the details of ADCs and all these things, analog to digital converters, but just a very basic thing. Listen, when yellow cell interacts with laser, this scattering is recorded by yellow cell is recorded by computer. Now computer send back signals to these electrical circuits. Computer now send signals to these electrical circuits that this is a yellow cell, therefore you must take positive charge. That means these are now positively charged. They are now positively charged, and when this cell comes, this droplet as a whole acquires, this becomes negatively charged. This becomes negatively charged. This becomes positively charged. I'm confused at particular times when I am talking about this, because you will know just after 2 minutes. So this becomes positively charged, and this droplet surface, listen, this droplet surface becomes positively charged. Okay. Now another thing is very important here, that is separating plates. What are separating plates? Now listen, suppose there are 3 collecting areas. Now listen, suppose this plate is connected to anode, this plate is connected to cathode. Am I right? Which one is cathode, which one is anode? Is this cathode or is this anode? What do you mean by cations? Cations means positively charged or negatively charged? Positively charged species, positively charged ion, positive charge move towards positive, they move toward negative. That means this is cathode now, this is anode. Anode is always positive because it attracts anions. Anions is negatively charged. Any electrode which attracts an ion, that's why anode. Anion plus electrode, anode. Okay. Now listen. So this is your what? This is your anode. Now listen. This is your cathode. Now listen. The potential difference between these two plates are 60,000. It's a very big potential difference. Why it is important? Because now listen. This cell, when it is coming here in this particular electric field, this is electrostatic field. What do you mean by electrostatic field? The field or potential is not changing at any place. It's a uniform potential field. It's a uniform electric field. That means any work done through this is zero. Any work, if you want to take any charge from here to here, the work done will be zero. Okay. Now listen with all your attention. So when this particular droplet is coming here, okay, so it will be what? Pulled towards this plate, right? That means it will come here. That means yellow cells, in any case, whenever yellow cells will fall down, it will always be collected here. Clear? Now listen, we have already told this particular system that yellow cells should be positively charged. That's it. That was our only motto. We have only gave this data to computer. That yellow cells should have positive charge in its droplet. That is the only thing. Now listen, in practical practices, it is very important for you to understand that how will a cell sorter know which one is yellow, which one is red cell, which one is blue cell. That means what I am asking is, how will a cell sorter, there is no such cells like blue, yellow cells in your body, right? You are not extraterrestrial animal, unlike some. Okay. Example is sitting right in front of us. Now listen. But point you need to understand is this, that how will a cell sorter know that a cell should be particularly charged with positive charge? How will a cell sorter know that a cell will have to be charged with negative charge? Or how will a cell sorter know that a cell should be uncharged? That is my question. How will this know? How will they know, right? Listen, any cell doesn't have any color, but we can impart color to a cell by adding an antibody whose FC portion has an additional fluorescent tagging, FITC or something like that, right? Right? That means we will have to tag each cell with an antibody. Suppose, so this is the real diagram. All the cells are actually of same color. For your experimental purposes, for your understanding purposes, I have done that. But actually, all cells are what? Of same color. But the antibody attached to the cell surface marker of each cell. We'll talk in details. Don't worry. The antibody attached to the cell surface markers of each cell have different what? Fluorescent proteins or fluorescent tags. So what we are actually telling computer, we are actually telling computer that whenever you see any cell having an antibody with an yellow fluorescent protein, add positive charge to it. So it is coming here. Suppose these are 3 WBCs. One of the WBC is having CD28. Suppose one of the antibody is having CD28. One of them is having CD4. One of them is having CD8. Suppose we have tagged CD8 antibodies. So we have tagged CD8 proteins with antibodies having blue tags. Suppose we have actually tagged CD4 proteins with antibodies having red tags. We have tagged CD28 proteins with antibodies having yellow tag. Okay. Now we have told this computer that whenever you are actually seeing any cell having what we have actually told to the computer is, whenever you are interacting with a cell which is giving out yellow light after interaction with laser, it should be charged positively. Its droplet should be charged positively. We have given instruction to the computer that if you encounter any cell having blue colored antibody or blue tagged antibody, then you should impart negative charge to the droplet containing the cell having CD8. Now we have already told computer that if you interact with any cell having antibody whose FC portion is connected to a red fluorescent protein, then you must not charge it. That means here you will get CD28 positive cells, here you will get what? CD8 positive cells, right? And here we will get CD4 positive cells. This is how you separate different kinds of WBCs. Okay, now listen with all your attention. Very important point to understand. Listen. A very interesting point you need to understand that I am giving you an example. Okay, I am giving you an example. Now listen. Just apply your brain, nothing else. Listen. This is the population of what? CD8 positive cells, right? They are all negatively charged. They are all negatively charged. and it is CD28 positive cells. They are all positively charged, right? Now, if you are in this lab who is utilizing this type of system, now once you touch this thing, once you touch this, you will get a shock. Why? Because there are too many positive cells, too many positive charges. So you will get a shock because these charges are not discharged. There is charge in it. In this particular solution, there is charge. So in lab, what we do is we actually connect these tubes to, these tubes to ground. That's it. We actually connect these tubes to ground. So that charges flow to the ground. Clear? Now, suppose, going into bit details, there is a flaw in this whole discussion. There is one flaw, and I can tell you the flaw is not in this particular section, but the flaw is in this section, that is tagging of antibodies. Why? The flaw is in the tagging of antibodies. Why? I was talking about, listen, CD4, CD8, these are what? WBCs? They are what? T helper type 2 and T helper type 1. These are WBCs, right? They are WBCs, so, right? They are actually WBCs. Now listen, most of the WBCs have FC receptor. What? Yes, that's true. Suppose you take a random WBC, suppose this is a macrophage. Macrophage has FC receptor. They have FC receptor. That means if you add antibody, the antibody will get attached here. Most of the macrophage or most of the WBCs have FC receptor. Most of the macrophage or WBCs have FC receptor. So when you are dealing with, when you are asked to separate WBCs using cell sorters, first step you will have to do is, you don't have to add the antibody which is tagged with a particular fluorophore. First step is this, you need to add an antibody against this FC receptor. Not understanding? First you will have to add an antibody which is against this FC receptor. Okay? And this is, suppose your CD28 protein. Suppose this is your CD28 protein, this is your FC receptor. First add FC receptor and anti-FC receptor antibody. Anti-FC receptor antibody. First add anti-FC receptor antibody. Then in the second step, add the primary antibody to CD28. Then add secondary antibody. You know what is the difference between primary antibody, secondary antibody? What is the basic difference between primary antibody, secondary antibody? Primary antibody binds with the primary protein. Secondary antibody, this is the secondary antibody. Secondary antibody binds with the FC portion of the primary antibody. Right? This is the FC portion of primary antibody. This is the FC portion of secondary antibody. Right? Now listen. This is the primary protein, or this is the marker, cell marker. What is the marker? Marker is a specialized protein to which we generate antibodies. Means these antibodies are monoclonal. Secondary antibodies can be polyclonal. Why? Because FC portion in most of the cases is the same. But whenever I am talking about a primary antibody which will particularly associate with CD28, that's a monoclonal antibody. That's a very basic difference. So this is a monoclonal antibody, and the FC portion of this monoclonal antibody will be the site for the interaction of the secondary antibody. And to the FC portion of the secondary antibody, there is one fluorophore attached, a protein, which on interaction with a laser will give out either green light or yellow light or red light, depending on which fluorophore you are using. That's it. Clear? Okay, sure, sir. So this is the basic difference between a FACS machine and a flow cytometer machine. Okay. In the next discussion, we will talk about in details how we can analyze DNA content of a cell, or how we can analyze cell cycle progression using flow cytometry. Okay. Yeah.